So hi everyone, I'm Natalie. Sorry, I have a bit of a cold, so I don't have a mask on. Can you hear me in the back? Okay, perfect. I'm going to talk a little bit about microbiome in the context of mainly human health. That's what I do research on. I'm currently a PhD candidate in biomedical engineering at Caltech. I work in Weston and Margol's lab working on this. But I've previously worked as a lab technician at the ABE-FC program. I'm also a community college student that then transferred to UCI, and then I did my PhD at Caltech. So if your students have any questions sort of about that process, I'm also totally open to talk about how that was for me. Just reach out. I'll have my email at the end. So this is about a roadmap of sort of what the presentation will cover. First, I'm going to cover what is the microbiome, why is it important, how can we study it, what are the current challenges, and then sort of careers, future outlook, job skills that you really need to be in this field. And so first, we'll just start with what is the microbiome. And so I'm sure many of you guys have probably heard about microbiome papers. It's a very hot field right now. Basically, what it refers to is just the microorganisms that inhabit a certain location. And so for me, I study human health, and so these mainly consist of these 4 groups of organisms. We have archaea, bacteria, viruses, and fungi. And so in various places in your body, all of these organisms inhabit some niche. And so normally when they talk about human microbiome, they're talking about locations when they talk about a specific microbiome. For example, your mouth or your oral microbiome has unique taxa that aren't found necessarily on your skin. Your skin also has a unique set of taxa that aren't found in your gut or your digestive system. I specifically focus on the digestive system. This is where most of the microbes are found in your body. And so I specifically study this region, but other areas of study also occur. There are also, the microbiome has also been studied in the context of other organisms. And so, for example, right now, plant microbiome is a huge field as they're starting to study this in order to optimize agricultural production. And so I specifically don't work with this, but my sister does at UC Riverside. She's doing her PhD on plant microbiome. And so if you guys also have any questions related to that, I can direct you to her. Okay. Next is sort of why is the microbiome important? And so I'm going to start with 2 sort of historical examples. And so many of you might be familiar with Helicobacter pylori. So this finding won the Nobel Prize in 2005, where this researcher basically took a culture of Helicobacter pylori and drank it himself in order to prove that this bacteria causes stomach ulcers. And so this was really fundamental finding because in many of these patients, Helicobacter pylori is non-symptomatic, but in certain patients, it causes these gastric ulcers. And so this is a very great example of Koch's postulate, where it's basically one microbe causes one disease. And he could show that very definitively doing somewhat unethical decisions.
How did he fare?
Huh?
How did he fare?
So he very carefully picked a strain of H. pylori that he knew was susceptible to a certain antibiotic. And so once he started to have symptoms, he was able to very quickly administer an antibiotic, and he ended up being fine. But yes.
Children do not do
Yeah. So this is sort of a great example of like a very simple study where if you have H. pylori, you have gastric ulcers. And so it's very clear causation of the microbiome. Another example of this is recurrent C. diff infections. And so some of you might have heard of Clostridium difficile, a C. diff. It is basically a life-threatening infection where basically antibiotics aren't always sufficient treatment. And so these bacteria infect the colon, and you typically get this infection after you've already taken a strong dosage of antibiotics yourself. And it causes life-threatening dehydration due to extremely severe diarrhea. And so this can also result in colon perforation or distended colon, which could then require surgery. And so if it was just a bacterial infection, you could just treat it with antibiotics. It's not horrible. But the worst part about this is about 20% of the cases, after they're on antibiotics, they get recurrent C. diff infection. And then once you have recurrent C. diff infection, 50% of those population again gets recurrent infection. And so it's very difficult to prevent this recurrent disease. That is until they found this idea of fecal microbiota transplant, FMT. And so basically how the theory is that after you take a strong bout of antibiotics, you're basically wiping out all of your healthy microbes that inhabit your gut. And so that enables C. diff to form and colonize there, when typically that niche is already occupied by the microbes that live there. And so that's why it often happens after you take antibiotics. And so the idea is then, okay, what if I then, after I give antibiotics, I then reintroduce a whole healthy microbiome to then repopulate the niche so that C. diff doesn't get an opportunity to. And so that's basically what they did. FMT consists of taking a healthy donor who's not on antibiotics, they take a fecal sample from them, and then they have the C. diff patient, they give them this typical antibiotic routine, and then they transplant in the healthy donor's microbiome. And so that causes those microbes to colonize, so then C. diff doesn't have an opportunity. And so this was really exciting because it was shown to have a 92% success rate in preventing these recurrent infections. And so since then, FDA has now approved oral administration of this mixed community for recurrent C. diff infections. And so, yes?
I'm going to ask the obvious question.
Yes.
Yeah.
How do they prepare the fecal oral administration?
Yes.
I'm not doing that.
Yeah, right.
So I will preface this, we'll touch on this topic again, but I will preface that the donor screening is intensive. They have a huge panel of potential pathogens that they're screening for in order to make sure that this isn't causing a secondary infection. But I believe for the oral administration, they basically take the feces, they slurry it, they put it in a pill that is pH sensitive. It's a polymer that's pH sensitive, and so it specifically delivers once it gets into the SI tract, which has a higher pH than the rest of the upper GI tract. But yes.
I think I heard like the donors do get paid for it. I think.
Yes.
One job.
Yes. So I don't know if you've ever done a fecal collection, but having been in sort of this field, it is incredibly difficult to get people to collect their own feces. People are much more willing to undergo like an endoscopy or colonoscopy than they are to actually collect feces. But yes, you can probably get paid for it. So yeah, so this is super exciting that the field is sort of moving towards a therapeutic. But the human microbiome has been correlated with actually a huge variety of diseases. And so, for example, there's been studies that show that human gut microbes are associated with obesity. There's other papers that show maybe it's leading to neurodevelopmental disorders such as autism. There's a lot of research on how the microbiome impacts inflammatory bowel disease, which includes Crohn's disease and ulcerative colitis. There's also some correlations on how the microbiome might be affecting the development of type 2 diabetes and how type 2 diabetes might be affecting the microbiome. And this actually I found the most interesting. There's a lot of studies that show that your gut microbiome actually influences how you respond to chemotherapy treatment. And so there's currently a lot of research in here on how we can modulate the microbiome to make cancer therapies more effective. Unfortunately, for the two previous examples I showed you, we have a clear causative agent. But for a lot of these studies right now, we're sort of in the correlative phase. And so we're still trying to figure out what is the causative microbe and how are these hosts and microbes interacting with each other. Okay, so next is sort of how can it be studied. And so this is a pretty standard framework of how a microbiome study might start. So you typically start with your microbiome data acquisition, and so this is identifying a cohort that you're interested in studying, identifying the right samples to study, and then obtaining the sort of microbial data information, which I'll cover next. Once you have this information, you can then find associations between the microbiome and some sort of host phenotype, whether that be disease or clinical predictions of a disease. And so this is what I was talking about, where this is sort of at the correlative phase. Ideally, you then go in and determine causality, and so this typically involves using mouse models, synthetic communities, human organoids, something where you can show that this microbe causes this disease. And then ideally, the last step is translating this into human therapeutics. And so this could include whole community transfer, which is basically what FMT is, or it could be using specific depletion of your pathogenic microbe or specific introduction of a helpful probiotic. going back to how the microbial data is acquired, there's many forms of it. I'm specifically going to focus on the sequencing because that's what I do. But basically, you can take a microbial community and obtain metagenomics, which is characterizing all the DNA that's present, or you can do metatranscriptomics, which is looking at everything that is expressed at the RNA level, or you can be looking at metaproteomics, which is then seeing what proteins are present at that time period, or metabolomics, which is looking at what metabolites are actually being produced by this microbiome. And so you can imagine this is actually going to become a huge data set, and so a lot of microbiome is working with multi-omics data, which means a variety of data types in order to try to understand what's occurring. And so, as I mentioned, I specifically focus on metagenomics, and so there's two main types of sequencing you can perform. One is this marker gene survey, which I'll cover here. Some of you might have heard of 16S rRNA sequencing. This has really been the workhorse of the last 10 to 15 years in the microbiome field. Basically how this works is all bacteria and archaea, and your mitochondria, have this 16S rRNA gene. And in this gene, there's regions that have variable sequence in green, and there's regions that are conserved. And so in your experience of doing PCR, this is actually a perfect place to design primers to these conserved regions that you can then amplify up, and then you can sequence the variable regions. And so this is known as amplifon sequencing, where you're sequencing up a specific gene in the genome. And so that's exactly how our 16S rRNA sequencing works. We design primers to the conserved regions, and then we sequence the variable regions here. And so once you have this sequence, you can actually use this to track phylogenetics. And so using the 16S sequence, you can then put it on a phylogenetic tree. This is showing bacteria and archaea, and show where your taxa, the organism you have, lands on here, in order to give you some identification of what it is. The main limitation of this is you are only sequencing a single gene in the entire genome. And so if you're really interested in looking at the function of this microbe, having a name may not be telling you what metabolite am I producing. And so that's when we get to whole genome sequencing, or WGS. I'm specifically going to cover shotgun sequencing, but there's many types, and if you're interested, feel free to ask me after. But basically how this works is you have your genomic DNA, you then chop it up into little pieces, and then you sequence each of these little pieces, you assemble them together into these contigs, and now you have reconstructed the genome of the organism you're interested in. And so if you're doing this, you can actually characterize all the genes that are present in that organism, so you have a better idea of what it could be doing and how it could be affecting disease. And so that was what my first paper really focused on in grad school, was we developed a method in order to do whole genome sequencing in samples with high non-microbial DNA. Basically, since shotgun sequencing just takes all the DNA that's present, if I want to study, let's say, an intestinal biopsy, I have a bunch of tissue, which means I have a bunch of host cells. If we look at this intestinal biopsy, typically the ratio is about 10 to 1 human to bacterial cells. And so already 10% of my reads are going to be bacterial, 90% are human. And then a human cell has about 1,000 times more biomass than a bacterial cell. And so if you really want to study the microbiome doing whole genome sequencing, you have to find a way to get rid of the human DNA, or you are stuck with only having 0.01% of your reads are useful. And so my method, the method we worked on, was able to deplete this human DNA so that we could actually characterize the microbial strains that inhabited your GI tract. Okay. So, any questions before I move on?
Is it possible to separate the microbes first before you actually disaggregate everything?
Yeah, so actually the way the method works is pretty simple. You could separate the microbes first, and that's how some methods do, like filtration and stuff like that. The problem is when you're getting a tissue, it is a solid piece of tissue. And so you need some way of disaggregating all of this together. And so the method I developed was basically just mechanical lysis that selectively lyses larger cells, and so the microbes are left intact. It's okay. Pretty simple. Okay, and then next some challenges in the field. And so FMT is obviously very exciting, but as you brought up, there can be some issues with reproducibility. basically each donor has their own microbiome, and so you're putting in some unknown, undefined donor microbiome into a patient and then assuming it's going to work effectively every time. There's already been studies that show it's not the case. There's some effect that they've seen called super donors, where basically certain people can consistently resolve disease across a wide range of patients. And so it's totally unclear as to why that's happening. But the field generally doesn't like things that aren't reproducible. And so there is a push to move away from the FMT and move into a synthetic microbial consortium. And so basically, with the FMT, you have this whole community that you don't know what it is, but if you just need a diverse community in order to resolve disease, we can do that in-house. We could culture 100 different microbes and then give them to a person, and we know exactly what we're giving them. And so this is eventually the goal, and this is very in line with sort of the precision medicine, where you could also characterize the patient's microbiome, see what niche are they missing, and then try to fill that niche with a consortium. And so this is hopefully where the field is going.
Were the super donors, did they have a very diverse microgenome?
They do, yes, they have a very diverse microbiome. Yeah. But the FDA has also said they're a little hesitant to approve FMT because of all these safety issues, and I mean, you are just taking a whole piece of tissue from someone else and giving it to another person. So there could be viruses in there and other things that you're not characterizing. So. Okay. One of the other challenges actually just happened this summer. Basically, bad science leads to long-lasting repercussions. There was a paper in Nature Methods that was published a few years ago that was recently retracted this summer. And so that was due to this paper, which found major data analysis errors invalidate cancer microbiome findings. And so these authors found that the data was improperly handled and that the machine learning programs were not validated. And so basically, this study had previously the original paper led to more than a dozen subsequent studies that now have to go back and revalidate their findings. And the original research group had a startup company based on this idea that generated millions in investment. And so this is a huge problem in the microbiome field, unfortunately, where some researchers, because this may be such a hot field, they might not be taking the correct precautions when they're processing their data and interpreting the results. And so this is just something to keep in mind that every time something like this happens, it sort of causes a little bit of discredit to the field, and so it's really important to be very careful with how you're doing your data analysis and interpretations. Yes?
Is it extremely rare, or?
It's not extremely rare as kind of paper retraction, but I think one of the main problems was it was in such a big journal. This was Nature, and so a lot of papers would then stem from this. But yeah, there is a website if you're very curious called Retraction Watch, where they basically report every time there is a paper retraction. Ideally, it doesn't happen in these high-profile journals that frequently. Okay. Cool. And then the last is sort of about careers and relevant job skills. I think the microbiome field is extremely interdisciplinary, and so there's a wide range of jobs and skills that if you're interested in having, you could apply it to the microbiome. And so obviously microbiology, molecular biology, genetic engineering. I did a ton of next generation sequencing, imaging, chemistry, statistics. As I mentioned, this is a lot of big data, and so computer programming, data curation, machine learning, and incorporating AI is going to become increasingly important in the future. And then here's a list of some of the careers involving microbiome research. So there's microbiome scientists, clinical trials. There's a lot of microbiome therapeutics currently in clinical trials, just not yet approved. But you can read through these. And then let me know if you have any questions. I'm happy to talk more extensively about the research I do right now. I currently work on sort of a different topic. I work a lot on human virome, understanding how the oral and gut microbiome are connected, and Parkinson's disease. I'm also happy to help out with scheduling a lab tour, or if any of your students have questions about grad school, feel free to let me know. And then Caltech also has a series of internships. I've mentored a wave and a surf student before, and so these are great opportunities.
So there's actually two that I looked up for high schools. So SRC is for any K-12 teacher from anywhere and Pasadena High School students. But SSP is also for high school students, okay? And then the others are for college students.
Yes.
I see that you have Parkinson's. Does Parkinson's disease relate to any of the gut microbiome, or is that just a very separate?
No, it relates.
Okay, that's what I thought. I just wanted to double check.
Yeah, I'm happy to discuss it. There is another researcher at my campus where we collaborate a lot with, Sarkis Mazmanian. He works a lot on something called the gut-brain axis. And so there's basically this idea that your gut can directly influence your brain, and your brain can directly influence your gut. And so he's done a few studies in mouse models where basically they found that if you introduce this specific bacterial strain, you actually cause worse Parkinson effects. And in their Parkinson mouse models, they don't have any bacteria, they actually don't develop disease. So there's clearly a connection, but they're still, I'm happy to talk about it, but they're still figuring out what it is.
And there's a free webinar, first series of workshops on brain-gut association for teachers. So I will send that to you. Or it's in the science events. That's very interesting.
Yes.
What would you recommend to eat? Was it your knowledge?
It's a great question. Yeah, I would say actually for probiotics, the idea is that your niches are basically already colonized. And so unless you're taking antibiotics, the probiotics don't necessarily have a place to colonize and stay around. And so I'm actually a little skeptical about the impacts of probiotics. There hasn't been a lot of research in sort of long-lasting effects of probiotics. A lot of them have sort of been like while you're actively drinking it, you know, but that could also be in the bacteria in the culture are producing metabolites that you're using. So I'm not sure. Yeah, there's been a lot of research on sort of like the diets that cause the highest longevity. And so there's been a lot of research actually showing that Mediterranean diets are able to produce a more healthy microbiome. It's very difficult to do diet studies, as you can imagine. It's incredibly difficult to have someone commit to eating something and only that for many weeks at a time. And so the diet studies are just quite challenging. Yeah.
Sorry. I mean, this is kind of goes beyond what you're studying. So just this last week, I had a alumni come and speak because he had an internship through City of Hope, and it was great. It was awesome, exciting. He was doing something about like ovarian cancer, but then I was a little bit like I felt like it was like a punch to my gut because he was saying that the researchers there were discouraging him from pursuing research because they were saying something like, oh, it's not what it used to be. And anyways, because this student was really excited to get into science and, you know, learn more, but then he goes to this internship, you know, working with researchers, and he comes back, and I think he felt almost like discouraged altogether. So I feel like, oh my, like that was kind of like a punch to the gut, because it's like, what? Just, you know, they can be honest, but I mean, you know, since you're in the field, I wonder if there's something maybe you could share in your experience that I can also bring back to my students to kind of, because I understand it's hard, it's like grueling work, but you know, maybe from your perspective, something that I can tell students that come back from something like that and just to encourage them. I'm not sure, you know. So.
Yeah, I can maybe add a little bit of advice. I do feel like everyone's experience at grad school is so defined by the lab that they're in. When I was picking a school for grad school, I was given the advice that your priority should be your PI, the people in the lab, and then the project. And so it could be that maybe that lab isn't necessarily a good fit for him, because I see that a lot in my grad school. We get a rotation, and it's like, oh my God, this is so not going to work out with my boss. But it ends up being fine in another lab. And so it could just be like a bad batch. I think a lot of the purpose of a lot of these internships is actually to figure out what you don't want. I think that's like the most important thing. And so you've tried something and you're like, I know this does not work for me. And then you can take that and apply it to your next avenue and then see if you see those same things, you know this isn't going to work out. That's my 2 cents, but sorry.
Can I interrupt you? So I would also say, what kind of research was he talking about? Was it at a hospital setting, or a university setting, academic setting, or industry? Because industry also hires a lot of researchers as well. Yeah, and there was a slide that Natalie didn't put in, but and by 2030, they're expecting $30 billion industry in microbiome therapeutics. So I think that is a job opportunity for our students, right? Yeah. So.
Any questions? I will say I do think the microbiome, echoing her point maybe a little bit, the microbiome is definitely sort of in the research stage. And so I do think over the next few years, as we start to learn more, we can get to the therapeutic level. But currently, as it is right now, there's very few diseases that we're directly treating with microbiome, such as the recurrent C. diff infections.
Thank you.